Telomerase activation therapies represent one of the most ambitious frontiers in the quest to slow or even reverse aspects of cellular aging. At the heart of this strategy lies telomerase, a unique enzyme capable of extending telomeres by adding repeated nucleotide sequences to the ends of chromosomes. In most somatic, non-reproductive cells, telomerase activity is virtually silent after early development, leading to gradual telomere attrition as cells divide. But in certain cell types, like stem cells and germ cells, telomerase remains active, allowing these cells to maintain their replicative potential for longer periods. The idea behind telomerase activation therapies is to harness or replicate this same rejuvenating mechanism in ordinary somatic cells, potentially offsetting the decline associated with age and chronic disease. Scientific advances over the last decade have made it increasingly plausible to upregulate telomerase in targeted ways. One of the most studied methods involves enhancing expression of the human telomerase reverse transcriptase (HTRT) gene, which encodes the catalytic subunit of telomerase. By stimulating HTRT, researchers can potentially restore telomerase activity in aging cells, thereby lengthening telomeres and improving cellular function. In animal models, such interventions have yielded impressive results: extended lifespan, improved tissue regeneration, and better metabolic profiles. For example, mice treated with telomerase gene therapy showed delayed onset of age-related diseases without an apparent increase in cancer incidence, particularly when therapies were applied transiently or confined to certain tissues. These preclinical outcomes lend hope that carefully regulated telomerase activation could translate into meaningful health benefits in humans. The potential benefits of telomerase activation extend beyond merely prolonging cell life. Longer telomeres are associated with more robust immune function, improved skin and organ integrity, enhanced mitochondrial activity, and even better cognitive performance in aging populations. In regenerative medicine, telomerase activation holds promise for helping tissues repair after injury or illness, such as in cardiovascular disease or degenerative neurological conditions. Some researchers have even proposed combining telomerase activation with stem cell therapies to amplify their regenerative capacity, potentially transforming outcomes in otherwise irreversible diseases. Although concerns around the cancer-promoting potential of telomerase reactivation remain valid, it's important to note that most recent studies are moving toward nuanced and highly controlled methods of delivery. The field has shifted away from blanket upregulation and is now focusing on localized, time-limited, or conditionally activated systems. Gene editing platforms such as CRISPR/Cas9 and advanced viral vectors now allow for precision targeting of telomerase activity, limiting it to specific cell types or activating it only in response to certain cellular signals. This evolution in therapeutic strategy significantly reduces the theoretical risk of inadvertently encouraging malignant transformation, which was a major limitation in early conceptual models. Additionally, naturally derived telomerase activators, compounds like cycloestragenol, found in certain herbal extracts, are being studied as gentler, low-risk interventions that may mildly enhance telomerase activity without the need for genetic modification. While these compounds are still under investigation and should not be overstated in their effects, their relative safety profile and accessibility make them an intriguing bridge between pharmaceutical-grade therapies and wellness-oriented approaches. Telomerase activation is still largely in the experimental phase when it comes to human application, but the direction of current research is increasingly optimistic. The scientific momentum, combined with improvements in molecular targeting technologies and risk management, positions telomerase activation as a powerful candidate in the arsenal against aging and chronic disease. Rather than simply extending the number of years we live, these therapies aim to improve the quality, vitality, and resilience of those years. As our understanding deepens, it becomes more plausible that carefully modulated telomerase activation could one day form a cornerstone of preventive and regenerative medicine.